in Terengganu. Okay, so you have to travel all the way to Terengganu if you like that. Unless you don't like traveling lah, you know, stay here only lah, you know. If you enjoy traveling and see places and at the same time find out what other states are doing in relation to live food production, then one of the trips we plan to do, once we are allowed to do so, is to visit the Tanjung Demong Research Station, and there you find you know, how they actually grow fish, marine fish into brood stocks, and then how they culture live food organisms like microalgae, rotifer, copepods, and so on lah. And we normally go visit farms which actually grow live food organisms to feed their to nurse their fish. Okay, all right. So those are the things hopefully we will experience, and hopefully the you know, we don't have any more lockdowns because of this COVID thing. It's so different, you know, when you go and visit places and see things for yourself. It's so different compared to just seeing videos and listen to talks online. Okay. All right. Now, just to be sure, are all of you in AKU4201 also, or only some of you in that particular class? Semuanya dalam AKU3201 juga ke? Atau ada yang hanya kelas ni aja, bukan AKU3201? Just to know. Ada? Atau semua di AKU3201 adalah di sini juga? Ya.
Rasanya semua je lah.
Iya ke? Wow, okay. Alright. Okay. Good. Now, lagi satu, another one. Okay, where's Atira? Atira mana? You there?
Here.
Oh, there you are, Atira. Atira Usna. So, in this particular class, AKU4402, Atira is your, what do you call this? Is my assistant lah, basically, and she would be in charge of your, will write down your attendance. Okay, Atira, is that okay with you?
Okay.
She's the class monitor. The other class, 3201, is somebody else. Okay, Ellie is the monitor for that one. Yang ini Atira. Okay, Atira, ya? So she will help me with your attendance. Okay, for the kuliah, for the lectures, and also the lab lah. All right. Okay. Now, I think I have 35. Okay, 34 of you, fine. That's good enough. Then I think I better start lah. Yeah, I better start. But anyway, before I do anything, I want to take a picture of everybody. As I said, right? This is for my record. Okay, so that when Atira compiled the list of attendees, I said, okay, just to make sure you guys are here. Okay, all right. Can you please smile, everybody? Senyum sikit. Late in TGIF, man. Thank God this Friday, all right? So, besok cuti. Okay, so I'm a bit crazy sometimes, so guys. Is it okay with you guys? Okay,
Okay je lah.
Oh, tak boring aja lah kan? Okay, so let's take a picture, everybody. All right. Let me see, hold on. Where should I press? Okay, there you go. 1, 2, 3. 1, 2, 3. There you go. All right, yeah? Just a quick one. Quickly, yeah? Yeah, I think I've got everybody's very beautiful, pretty, handsome faces there. All right. Okay, let me save this for a while. Save. Screenshot desktop. Okay. Right, got that. Okay, now, Lo, Meng Fong, how are you, Meng Fong?
Yeah, good.
Yeah, I tengok you macam, actually you remind me of this Kung Fu movie, you know, Kung Fu movie. Your baju are green and yellow in color like that, and then, you know, I can see your hair very short. Or is it your hair short or you actually, you know, ikat belakang?
Yes, I cut short.
Ikat short, no wonder lah. In the 1970s, there was a movie called Kung Fu. Do you guys know this? Maybe you guys are too young. During those, you ask your dad lah. You ask your dad or your mom. In the 1980s, there was a movie, or late 70s, there was a movie called Kung Fu, and the Kung Fu Sifu, in the movie was not Chinese, he was American, but he made his head bald, all that, like a Kung Fu master. Okay, and the Shaolin at the Shaolin temple, the master there called him Grasshopper. So you look very much like that particular character called Grasshopper, All right. Just for having fun lah, Meng Fong. All right. So you Google that one, Kung Fu, and then you see how the Kung Fu master talk to the Grasshopper guy. So anyway, I just like to talk to you today. It's a live food production class. So before I go to that, I just want to ask you all, in your mind, when you talk about live food, what do you have in your mind? What do you think about? Dalam fikiran you all kan, bila kita cakap makanan hidup or live food, apa yang timbul dalam you punya fikiran? Mashita, can you say something? What do you understand by live food organism? Mashita binti Shah?
Ya, saya.
Yes, Mashita. Syarudin. Yeah. Kalau I cakap live food organism, what comes to your mind first?
Makanan yang sihat mungkin.
Sihat mungkin untuk apa? Untuk siapa?
Untuk animal. Carnivore semua.
Makanan yang sihat untuk animal, right? Okay. Gabriel, what do you think? What comes to your mind first, Gabriel, when you say about live food organisms?
Yang banyak protein
Yang banyak protein. Okay, Nur Saada.
atomnya.
Nur Saada.
Makanan yang directly dia hidup dalam, contohnya dekat dalam lautan macam plankton.
Macam plankton hidup, yeah. What about you, Tan Hong Sheng, Tan? What do you think?
Sebenarnya live food organism boleh cakap ialah makanan yang boleh kepada ikan dan juga kepada kita manusia.
Okay. Alright. So Suhada, what do you understand by a live food organism? Ada fikiran you apa dia? Suhada.
Makanan yang berasaskan tumbuh-tumbuhan.
Makanan yang berasaskan tumbuh-tumbuhan. I mean, food for fish which is based on plants, is it? All right? Okay. That's part of it, all right? Evelyn, what do you think, Evelyn? Evelyn, are you there?
Yeah.
Yes.
Bagi
Yes.
saya makanan live food organism ni adalah makanan yang diproses untuk ikan dan juga manusia yang boleh makan, sama ada tumbuhan akuatik ataupun plankton.
Okay. Ikan, makanan boleh makan oleh ikan yang diproses. Okay. Okay. So kita kena we have to understand the word process there. All right. It's all right. Okay. What about you, Norain Natasha?
Makanan hidup untuk bagi makan kat ikan.
Okay. Ikan saja ke atau lain daripada ikan pun boleh? Only fish or other than fish?
Hidupan akuatik.
Okay, that's better. That's better. Hidupan akuatik. What about you, Chu? Chu Lip Ching? What do you understand by live food organism? Or tiny blur sikit? It's okay. If blur also no problem, man.
Live food organism is like
to give the fish having the food, so like make the fish food like that.
Never mind. It's okay, Cho. Don't worry. Don't scratch your head so much. Scratch your head so much, your hair also fall down. Huh? Yagis, what do you think, Yagis?
Kind of like produce or either processing like food for only for aquatic animal. It's including fish, crustacean, mollusks like that.
All right. So you guys already mentioned some things there. Quite close. There are some points that I mentioned. You give food to your fish. Ryan say give food to aquatic organisms. In fact, that is better. Huh? It's better to use the term aquatic organisms or aquatic species. Yeah, yang where we could want to give the food to. And then the other thing is that one of you mentioned about aquatic plants or phytoplankton. Yes. And we should also include zooplankton. All right. And when you say phytoplankton or zooplankton, these are usually, oftentimes they are what we call as planktonic in nature or microscopic, meaning you want to see the details of all these organisms. You need to look at them under the microscope. Okay. So in this particular class, even though the term live food organism is very wide, meaning that anything also which is alive given to aquatic species, but during the course of our study, dalam kita punya pengajian dalam kursus ni, we will focus on the species of plants and animals, which are mostly very, very small size in nature. Most of them are microscopic. Okay. And then these are the ones that we tend to give to fish. Now, when I say fish, it includes also mollusks, crustacean, okay, and all those aquatic organisms, which you must give feed to. If not, they will not grow. All right. So essentially, why don't I just start off with my slides so that we can go quickly and explain to you a bit more detail, the meaning of live food organisms and thereafter the remaining slides. Okay. Okay, can you guys see this? Boleh, ya?
Yeah.
Good. All right. Thank you. Okay. So, oh yeah, by the way, this is SpongeBob. This is one of my favorite characters lah. Okay. you guys know me. I just tend to have fun with my students, and so this SpongeBob SquarePants is one of my favorite characters. In fact, I prefer his other friend. You know who his other friend is? His close buddy.
Patrick.
Oh, Patrick. Yeah, I said Patrick. Patrick the starfish. All right. And we don't, unfortunately, I have not seen people growing starfish. But starfish and even this porifera or sponge, no. SpongeBob is a sponge, of course, and they consume live food organisms in the at Bikini Bottom, you know. All right, so you go through the series, you know, they live in this place called Bikini Bottom. But interestingly, in Bikini Bottom, there's a desert there. There's cactus growing, you know, fantastic. Anyway, one of the things that you need to be aware of, because you guys are involved in aquaculture, right? You grow fish, and you don't want your fish to fall in love with your food. Okay, you want your fish to eat their food so that they can grow. Yeah, not that if you put your feet in there and the fish say, this is so lovely, I fall in love with my feet, I just want to keep it here, I don't want to eat it, then they will slowly starve to death. All right. So one of the things you want to make sure, regardless whether you give live food organism, sama ada kita bagi ikan tu makan makanan hidup atau makanan diproses, jangan biarkan dia jatuh cinta dengan makanan sebab dia tak makan langsung. Matilah dia, mati kebuluran. Yeah. Alright. Okay. Anyway, when you talk about live food organism, okay, this is basically a definition which encompasses what we talked about just now before we start this slide, right? And some of you mentioned a couple of these things. Live food organisms include all plants, okay, in bracket phytoplankton lah. Why phytoplankton? Because majority of live food organisms are in planktonic, okay, and animals, zooplankton, which are usually cultured separately and provided alive. Now, this is important. Provided alive, they are given alive as food to economically important cultured aquatic species. So then you have a situation where these live food organisms, you culture them and you grow them separately in maybe a container somewhere, in tanks, in ponds, all right? So you grow them and then you collect them, you harvest them, and you have to give them to the fish that you are culturing, all right? So this is a bit interesting in terms of aquaculture, because normally you would give your fish pellets, right? Fish pellets. But when they are very, very small, some of these fish in the, we call it larvae or fingerlings, you know, they don't even want to eat the pellets. When they eat the pellets, they might die because the pellets do not have the amount of nutrient which is required by the baby fish, okay? They may not have the enzyme to digest the processed feed. So therefore, they depend very much on the presence of live food organisms. Okay? All right. So this is basically a definition that should include a lot of the things we will be discussing as we go along during the semester. So, but we'll focus basically on all these planktonic species, yeah? All right, so when you talk about aquatic species, now aquatic species can be your fin fish, fish with fins, such as your grouper, such as your sea bass. It could be your mollusks, such as your oyster. Okay, it could be mussels. It could be crustaceans, like your shrimp, like your crab, okay, and so on. So you are growing this live food organisms, either plants or animals, to feed to the animals that you are culturing. Now, brief history about live foods. Now, initially in the 70s, in the 70s, those days I was still very, very small. I was in primary school in the 70s. We found that the business of aquaculture is very new in Malaysia. People just started to grow fish because mostly during those times, we catch our fish from the sea, all right? We catch them from the sea, from the seashore normally, all right? Sometimes go a bit deeper. And you know that in the 70s, there's a lot of fish that we capture. Nowadays, the fish capture is dwindling already down. If you've taken my, in my AKU4402 class, you can recall that now our capture fishery is going down. The trend is going down. Okay. Okay, if we don't do anything about it, we can't even have enough food that we capture from the sea. But those days, in the 70s, yes, we capture a lot of our fish from the sea, and we also capture their fry, their fingerlings from the seashore, right? But when the aquaculture business grow, when the industry expand, when the demand for seafood increase because of more population in Malaysia, we find that we don't have enough fish to be captured from the seashore. Apabila permintaan kepada ikan bertingkat, bertambah, maka sumber benih ikan ni sangatlah penting. Okay? Kita tak boleh lagi mengharapkan penghasilan benih ikan atau untuk kita besarkan atau kita tangkap daripada pesisiran pantai. So apa kita nak buat? What do we need to do? Okay, so what we need to do is to have hatcheries. So in the 1980s and the 1990s, we saw a proliferation or an increase in the number of fish hatcheries, shrimp hatcheries in Malaysia. Pada tahun 80-an, kita lihat ya, banyak orang mula menternak ikan dan keperluan benih makin meningkat juga. Oleh itu, kita lihat banyak hatchery dimulakan atau dibangunkan pada tahun 80-an dan 90-an. During those days, in the 80s and 90s, we find that lots of hatcheries were being developed. I was involved during that time. I started involvement in the fish hatchery business in the mid 1980s. All right, we grew shrimp. We grew tiger shrimp those days. All right, and one of my role was to actually grow them in the hatchery. Okay, and to grow out as well, production. So, when you want to grow shrimp in the hatchery, when you want to grow fish in the hatchery to meet the requirement of the population, to meet the demand, one of the important things you need to do is to make sure you provide enough food for the fish larvae, or fish fingerlings, and so on. Okay, to provide enough food, you can't simply give them processed feed. You can't give fish pellets to the fish fingerling or fish fry or fish larvae. You must give them live food organisms in most cases. Now, let me say this again. Not all fish larvae, okay, require live food. Okay, ingat tak semua ikan, larva ikan atau fingerling atau fry ikan yang mesti dibagi makanan hidup. Setengahnya boleh terima makanan rumusan. Remember this, Not all the fish larvae, fingerlings or fry must be given live food. Some of them can be given processed feed also, depending on the nature of the larvae. We will talk about that later, okay? Now, enhanced production of high quality and healthy larvae is the key target for successful and competitive expansion of the aquaculture industry, okay? And also the issue here is a lot of problem with survival rates, variable growth, and so on. Now, normally, we find that the preferred live food organisms for larval fish are those in their natural diets. Okay, this is a very important point, that the arrow number 2, The preferred live food organisms for larval fish are those in their natural diets. Maksudnya di sini, makanan untuk larva ikan yang biasanya, okay, dia perlukan, ya, yang dia perlukan biasanya adalah jenis yang dapat di alam semula jadi, di mana mereka dibesarkan. Alright, so that is the preferred food organism. For instance, if the larval fish is in the coastal area, near mangrove areas, and they feed on rotifer, they feed on copepods. These are two important live food organisms, rotifer and copepods. If the larval fish has been feeding naturally on copepods, on rotifer, when you grow them in the hatchery, they also would prefer those kind of diets. Okay, but they eat all sorts of live food in the natural system. Dalam sistem semula jadi, larva ikan ni akan makan pelbagai jenis makanan hidup. Betul tak? Ya, makan hidup pelbagai jenis. Kalau rotifer tu pelbagai spesies rotifer. Kalau copepod, pelbagai jenis copepod dan sebagainya. Tapi yang ini tidak praktikal untuk kita hasilkan dalam hatchery. We have a myriad of or so many live food organisms in the ocean that the larval fish that we want to grow feed on, but we cannot grow all of them. We have to choose, pick and choose. Kita kena pilih mana yang mudah untuk diternak, mudah untuk dihasilkan for our purpose. Alright? So, something which is commercially practical. And one thing that you may want to know also, why that we need to give live food organisms to some fish is because of this particular, what do you call this, point here, all right? The fact that some fish larvae, okay, when they have absorbed what we call as their yolk sac, okay, dia dah serap dia punya kantung yolka, they absorb the yolk sac, they still lack the digestive enzymes to digest protein in artificial feeds. This is very important to understand. Some fish, when they have absorbed their yolk sac, they are already able to take live processed feed. Some fish, when they absorb the yolk sac, they are still lacking the digestive enzymes to digest processed feeds. So what do? For this sort of fish, larvae, they must be given live food organisms. Why? Because one of the important aspect of live food organism is that they contain enzymes. They contain enzymes which can help to digest the what do you call this? The food already in the gut of the larval fish. All right. So this is provided also by the live food organisms, and meaning the live food organism is easily digested, and it can also trigger the larval fish to start making their own digestive enzymes. Okay. And the larvae of some species, shrimp for instance, change diet or change habit as they mature and acquire a change in food types. Somebody just joined, right? Let me see. Ahmad Samsudin. Okay. All right. All right, so live food organisms contain all the nutrients such as essential protein, lipids, carbohydrates, vitamins, or what have you, amino acids, which is the precursor of proteins, and fatty acids, and hence are commonly known as the living capsules of nutrition. Okay, and the success in the hatchery production of fish fingerlings for stocking in the grow-out production system is largely dependent on the availability of suitable live food. Important, eh? All right, as I said, remember, largely dependent on availability of suitable live food. Not all of them, but a majority of them require live food. All right, especially the marine fish. So what are the advantages of live food compared to inert diets? Okay, what are inert feeds? Inert feeds are processed feed or formulated feeds. So when you read any report or any slides which says inert feeds, formulated feeds, processed feeds, they are the same, meaning that you have to make them. Okay, so the opposite of live food is inert feeds, processed feeds, artificial feeds. All right, these are all we need to make. So what are the advantages of live food compared to inert feeds? Number one is that it swims in the water column, especially your zooplankton, available to the fish in many cases, better contrast and movement, and the fish will tend to find it more palatable and so on. All right. And not only palatable, it has, as I mentioned earlier, it has this digestive enzyme, remember? It has this digestive enzyme, especially for zooplankton, that can assist the very young fish larvae to, what do you call this? To absorb its feed, which is already processed. Now, for practical purposes, these are the terms which I like to share with you. What is embryo, which is developing larvae inside the egg. Number two is yolk sac larvae. Now, yolk sac larvae is what we also known as free embryo. It only hatches from the egg, but it has this yolk sac that got stuck to the belly, yolk sac. And once the yolk sac has been removed or actually absorbed by the growing embryo, it becomes a larvae. Now, there's a term here which you may not be aware of, or you may know. It's called exogenous feeding. Now, what is exogenous feeding? Exogenous feeding means feed from outside. Feed from outside. Exogenous feeding. Now, when you have the yolk sac, it is called endo. E-N-D-O. Endo. Endogenous feeding. So, endogenous feeding means larvae which consume food from the yolk sac, while exogenous feeding is food that you must give to the larvae. So this exogenous feeding is food that provided by the guy who feeds the fish. If you're feeding the fish, then you give the fish the live food organism or pellets, then that is exogenous feeding. Okay. Once from larvae, it becomes juveniles, fry fingerlings, where the form and function of the stomach is fully developed and so on, until adult, which is sexually mature. Now, endogenous feeding is normally up to 3 to 5 days, sometimes even 2 weeks for arowana. And then you have mixed feeding. What is mixed feeding? Mixed feeding means while the fish is consuming food from the yolk sac, you can already give it some food when the mouth is open. All right. So after a while, you feed it exogenously all the time. Okay. Now, two things which I want to share with you is this particular term. All right. Now, I must, what do you call this? Apologize for lack of pictures or any of the graphics. But later on, I'll show you some more pictures on this. But it's just some facts first for you, all of you. Some fakta dulu. So until this point, up to this point, what I'd like you to know is that, okay, we have these live food organisms, which we give to our fish, all right, that we are growing in the hatchery, especially in the hatchery, all right? So we are growing the fish larvae in the hatchery, and they must be fed. And we feed them live food organisms in most cases, all right? And the live food organism, we must grow them in separate tanks normally, okay? So up to this point. And then when you want to give them to your fish, what kind of fish that you should give them to? Well, there is two extreme of level development that I like to share with you. One is called precocial larvae, precocial larvae. The other one is called altricial larvae. So these two extremes. One is called precocial larvae. The other one is called altricial larvae. What are they? What are they? You know? Well, precocial larvae develop rapidly, have more energy. When the yolk sac has exhausted, the larvae appear as mini adults with mature digestive system and functional stomach. Okay. Okay, when you look at a fish larvae, then you know this precocial. When you see it, wow, look at the yolk sac, man. The yolk sac is very clearly is large, is yellow in color, you know. And then when you see that, it will tell you that that particular fish has a lot of energy available in the yolk sac, a lot of food available. And then after the fish has absorbed the yolk sac, it's a good chance that the fish can be given processed feed directly, given pellets. When you look at the next one, it's called altricial larvae, which is poorly developed in terms of the larvae, what do you call this, metamorphosis or larval development. When the yolk sac is exhausted, the larvae remain in an undeveloped state and lacking a stomach. Okay, or stomach is not fully functioning even. Now, in most cases, we find that fish which has precocial larvae tend to be greater in number. For instance, an example of a precocial species, all right, is ikan siakap or sea bass. Okay, for example, 1 kilo of ikan siakap, 1 kilo of sea bass, when they produce eggs, you can find maybe about 500,000 eggs. All right, now ikan siakap is an altricial species and producing altricial larvae. All right, so because it has very poorly developed yolk sac, developed fry or larvae, and yolk sac is very small. What is the trade-off here? Trade-off is they have to produce a lot of eggs. So ikan siakap, for instance, when you grow them up to maybe for a breeding size, let's say 2 kilo, example only, sometimes more, 4 or 5 kilo fish, a broodstock. They say you grow the siakap up to a breeding size. For every kilo of ikan siakap that you can weigh, broodstock, you can get maybe 500,000 eggs. Okay, but for tilapia, which is a precocial larvae, tilapia, for 1 kilo tilapia, you might only get about 3,000 eggs. Can you see the trade-off? So tilapia is a precocial larvae, all right? So when the broodstock produce eggs, it gives out about 3,000 eggs per kilogram. When you look at siakap, it is an altricial species, produce altricial larvae. 1 kilo of siakap produce about 500,000 of eggs. Okay, so that is a bit different in terms of the number of eggs in relation to the larval development. Okay, now this, I hope this is clear. You can see this. Don't worry about this. I will share with you some slides later. The slides I will share with you in your WhatsApp, so that you can review that as well. When you look at this is altricial and this is precocial. All right. Now, altricial development hatching is limited. Fecundity is high. What is fecundity? Now, fecundity is the number of eggs per unit body weight. Number of eggs per unit body weight. For instance, eggs per kilogram of body weight. Okay. All right. So for artificial larvae, as mentioned just now, There are more eggs per kilogram of body weight for artificial, and precocial is relatively low. Example, tilapia, right? This one, example, sea bass. Yolk per egg is low, high, parental care, not much parental care. This is variable to a lot of parental care. Breeding type, pelagic. What's pelagic? Pelagic means in the water column, meaning no parental care. Nesting, like some of the bluegill, cichlid, brookmouth cichlid, just like your tilapia, and live bearing like guppy and shark and so on. You know that guppy, when they leave their mother's womb or comes out of the mother's what do you call this, body cavity, your guppy is already able to feed, you know. You can give it processed feed, okay, pellets, small size pellets they can eat already. Because when they go out of their mother's body cavity, they already absorb the yolk sac for guppies. All right. Examples, herring, cod, alewife for this one and so on. All right. So this is to show you the different characteristics or different attributes of artificial and precocial larvae. All right. Okay, you can read all through this. Okay. And artificial larvae must rely on feed source that are at least partially or easily digestible, large amount of amino acids, contains enzymes to help digest food particles, and supplies all the essential nutrients. Now this is a picture of Atlantic salmon. You look at this Atlantic salmon with yolk sac. Look at this yolk sac. It's huge, it's big. Big yolk sac, yellow in color, full. All right. Look at that. Atlantic salmon, look at the yolk sac, large and yellow in color, full of yolk. Look at this gillhead bream, very transparent. And you look at that, you find that there's very little yolk inside here. Look at this picture. This is an Atlantic salmon in the picture. What about the small ones? The small ones are the gillhead bream, just like this. And you look at this gillhead bream, number one is that it is very, very small. And you look at the yolk sac here, below here. It's almost transparent. Not much nutrient here compared to this one. So one of the characteristic of precocial larvae is that the larvae tends to be big and has a lot of yolk. One characteristic of artificial larvae is that the larvae tends to be quite small and very little yolk available for the growing larvae. So that is why it is a bit more complicated, it's a bit more challenging to grow marine fish larvae compared to freshwater fish larvae like ikan tilapia. All right, so there are reasons for this. Okay. All right. The transition between endo and exogenous feeding is critical and often accompanied by low intestinal enzymatic activity and also increased mortality and reduced growth. All right. Now, one of the most important aspect of this that we've gone through is this one, this point number 2. Fish larvae is especially vulnerable to malnutrition at the earliest stage of feeding. Now, let me give you an example. Let's say you grow your gillhead bream, for instance, the hatchery. Let's now mention gillhead bream. Okay, and then when it grows to it's growing to a certain size, you feed it with live food, right? But before you feed it with live food, the mouth must open. What do you call this? The mouth must be open first so that it can just grab whatever food is available to the growing larvae. So here you are. You have the gillhead bream with the yolk sac attached to it, and it's consuming the yolk sac, consuming until a stage where it says, "Okay, yolk sac finished. I must eat from outside. I must eat exogenous feed that given to me by my owner. So once the great bream has consumed all the yolk sac and the mouth is open and it is ready to feed from outside, and you do not give it the right amount of feed at the right time, it will die. So in the hatchery, the timing is very critical. Timing of when you should provide your live food organism to your growing fish larvae. Because if you give too early and the fish has still got the yolk sac, it will not eat the live food organism, and the water becomes dirty and polluted, and then you might have problem with your fish, your ammonia levels going up high and so on. If you feed too late, the fish will die, they get starved. So this is why in hatchery procedure, in hatchery practices, it's very important to make sure you provide your growing fish larvae, your growing shrimp larvae with the right kind of live food at the right time. The right kind, the right quantity, at the right time. Once you miss that window of opportunity, you don't give it the right food, it will die, and that's it. All right. At the same time, you must also remember the size of the food that you give to your fish must be of the size which can be digested or captured and digested by the fish. So remember, the fish will eat once it's consumed its yolk sac, it becomes hungry, it wants to eat, and you provide with the live food organism, right? But if the live food organism is too big for the mouth of the fish, it cannot eat, then it will also starve to death. So here you are, timing very important when you want to give. Number two, you have to make sure that you give the right size. Too big, it will not eat. Okay, it will get stuck in the mouth also. Okay, this is essentially the size and level length of fish and their hatching rates. Yeah, okay, don't worry about this so much. And then some comparison on between shrimp and fish. Shrimp is a bit different because you give the feed a bit larger. The shrimp has what we call as appendages, which can actually grab onto the feed and bring it to the mouth. So if a particular feed is of very big size, cannot be eaten directly by the fish, but maybe the shrimp can eat it. Why? Because the shrimp can capture the live food with its appendages, okay, with its claw. All right, so this is one fish, one species of fish which is quite interesting. What fish do you think this is? What larvae of what fish? Any idea? Anyone? Siapa boleh teka ni adalah
Arowana.
arowana? Good. Okay, good. This is the yolk sac larvae of ikan arowana. Okay, the yolk sac larvae of your arowana, and you look at it. See this? Look at the yolk sac, man. Huge, like a ball. Okay, it's even bigger than the fish itself. Look at the fish. Look at the yolk sac. Now yellow in color, a lot of nutrient. Okay, and you can see that even from this size, you can see the yolk sac very big, right? And it grew to up to this size. Even up to this size, almost a juvenile. All right, the yolk sac is still there, hanging on to the base of the fish here. Now at this stage, you will see that the fish mouth can open already. You know, this size, this stage. So in this particular stage, the fish can eat both ways. It can consume feed from the yolk sac. At the same time, the mouth can open also and take food from outside. So you have a case where you have here endogenous feeding plus from outside exogenous feeding. Now this is called mixed feeding, right? Mixed. Endo and exogenous feeding. All right. Okay, so just to let you know, okay, some fish, before they absorb all their yolk sac, they can open their mouth and ready to capture food which is available for them. Now, again, important of aquaculturists like all of you to give the right size of feed particles to your fish. This big fish, it's a medium-sized fish, all right, and in one gulp. And this medium-sized fish, it's the smaller fish in one gulp. Of course, the big fish can eat this very small fish, but you will need to spend a lot of energy catching this small fish and a lot of small fish to feed on. So if you feed your fish, make sure it can eat the fish, the fish that you give them or the live food, but make sure it's not too small or too large. Just the right kind of size for the big fish. Okay, all right. Now, these are some of the challenges of live food production. Before I go to this part, I like to take a break. Okay, let's take a 5 to 10 minute break, and I will come back and I continue with the other part. Is that okay with you guys?
Oh, kejap lah.
Boleh ya?
Yes.
All right, so why don't we take a break, and then if you have some questions for me as we go along just now, please ask me, and then I will continue to the next slide. Okay, let's take a 5 to 10 minute break, guys. Pick one. Okay. Okay, right. So as we said earlier, the live food is important for your level fish, and it provided some advantages, right? What are the advantages I mentioned earlier? Okay, example, it's moving. It provides some attraction to the fish larvae. There's good contrast. All right, it provides the required nutrition. It has enough. It also provides the digestive enzymes. All right, so those are the benefits of your live food organisms. But you must also make sure that you give the right size of live food. Right size, size yang betul. Too small, your fish will have to do a lot of work to find the food, and then eat a lot of them, which sometimes requires a lot of energy. Too big, it will not eat the feed, cannot feed the mouth, and it may end up polluting the water. All right, you have to give it to the right size. Okay. Now, these are some of the challenges. Number one is expensive. It's not cheap to grow live food, especially in hatcheries. You need to have specialized containers. So that is why, sebab itulah dalam mula-mula sekali kita punya takrifan, kita mula-mula definition of live food organisms. I mentioned that you grow the live food organisms, plants and animals normally in separate containers. You don't grow them together with your fish, Sometimes you do, sometimes, but in most cases, you grow them separately. So where do you grow them? In hatcheries now. That's why in hatcheries, you have special compartments or areas where you grow your live food, okay? Later on, as we visit some places, I will show you also how they grow live food organisms in those areas. I can show you some pictures as well later on, all right? And when you have to grow them in special areas or separate from the larvae which are growing, then becomes expensive, all right? And then you also need skilled and trained personnel, people like you, graduates of UPM, trained in aquaculture, skilled and trained to go out into the real world and contribute to the industry, right? So when you have already with you your knowledge, your information, your contacts, and then you go out, even though you may not have the skill required, you can understand pretty much how to get it done, all right? It takes time to grow live food, planning. You can't like grow your food today and tomorrow you want to give it to your fish. It takes time for the live food to actually, for instance, for some of your live food comes in capsules or in egg form, okay? And then it has to hatch first. It has to hatch and grow and you know, in enough abundance or quantity to be fed to your fish. So for it to hatch and to grow and to enough quantity, it takes time, all right? And timing. As I said earlier, if you can recall, between the endogenous and exogenous feeding, all right, there's a time in between called mixed feeding, right? Now, once your fish had absorbed the yolk sac, that is called, you know, the endogenous feeding part, phase, and ready to take the exogenous food, live food given to them, that live food must be given in time, all right? Too late, they will starve to death. So very important. That's why we put here critical timing. Huh? Now, when you want to select a live food, what do? Okay, when you know, live food for your fish. All right. So one of the important thing is that for you as a culturist, let's say you are running a fish hatchery. you graduated from UPM. You go out and you say, you know, I think I like to run my own fish hatchery. Okay. You said you want to run your own fish hatchery or shrimp hatchery. You dah graduate dari UPM ni, you keluar dan you ada keyakinan untuk menjalankan projek penghasilan benih ikan atau udang. Okay. So you want to do that, and then you need to have a space in the hatchery where you grow live food. Okay. So kalau you nak grow live food, what are the things you need to look at for the culturist, for you as the guy who runs the business? One is availability. Is it available in the market? Can you find the live food organism? If you cannot find it, what do? Let's say you want to grow a certain kind of microalgae, all right? And is that microalgae you want to grow to feed to your fish, is it available in the market? Do other people grow this particular microalgae? All right, can you buy from them the stock? Can you buy from, import from other countries, for instance? Okay, can you do that? All right, so sometimes when you want to start the growth of a particular zooplankton or phytoplankton, you might have to get the stock culture, or what we call as inoculum, the starter culture from somebody else. You might not have that yourself, you know. You might have to buy them. And you start growing them in your hatchery. Number two is cost effectiveness. Is it costly? Is it cheap to grow them? All right. If you can grow a particular live food, which is very good nutritionally, let's say a particular microalgae has very high nutrition for your fish larvae, very good nutrition, but then you cannot grow it in good abundance. You can't grow it well. Okay, it dies easily. It's very sensitive to the changes in temperature, pH, and light, and so on. So if you want to grow a particular microalgae, which is the best in the world, but then it is not cost effective, then you might opt to use something else. That's it. I mean, you know, you just cannot do it because it's so expensive. Number three is simplicity. What does it mean? It means that you need to find a particular species or particular food, live food, which is simple to grow, not complicated. Simple means like what? Simple means you have the, let's say you want to grow a certain zooplankton, you have the cyst or you have the egg, okay? You can incubate it in water in 24 hours, it hatches. After it hatches, it's already a good size to be fed to your fish or to your shrimp. That is simple, you know. Take the eggs of the zooplankton or the cyst, they call it, okay? You incubate it, and then within 24 hours, it hatches, and then you have the live food coming out, and you can already feed it to your fish. That is excellent. Or if you have another kind of live food organism, which when you put in the inoculum, you know exactly within 1 week how much is the density, and you can know how to calculate to feed to your fish. It's very simple, no? Even though you can, what you call, it's not so complicated. Versatility means what? Means that you can grow it in water with high salinity, low salinity, in fresh water, same species, okay, high temperature, low temperature, slightly higher pH, slightly lower pH. Okay, you can grow it in the cement tanks, canvas tanks, or you can grow it in fiberglass tanks, all right, or in ponds for that matter. So that is called versatility, very versatile. So if you have a live food organisms which is available to grow, it is cheap enough to grow, simple for you to grow, and versatile, then normally this would be a good choice for the culturist. All right. Now this is for you as a culturist, but for the larvae don't care so much about your cost effectiveness or your simplicity. They don't care so much about it. Okay, or they don't care about your versatility, how versatile it is. What the fish larvae wants are this. For the fish larvae, what it wants is availability. When it's hungry, alamak, I'm so hungry right now, I want my food. So it's available to them. Anytime the fish larvae wants to eat, it's available. Number 2 is purity. What does it mean by purity? Purity means no contamination. If you want to give it, let's say, a particular microalgae or mixture of microalgae, it should not be contaminated with zooplankton like ciliates. Okay, cannot be contaminated with what do you call this? Animals or what do you call this? Or bacterial contamination. All these things. Pure. All right. Number 3 is size. I mentioned this several times already, right? The size should be enough to be small enough for the fish to gape size or the mouth size. Acceptability, all right, you can accept the food. Maybe the food in terms of the movement of the food, the color of the food, all right. So all that attractive to the fish, it can accept it. And then digestibility, it can eat the food and then it can digest properly. Now remember, some microalgae are not as easily digested as others. So that's why some microalgae may not be useful for or may not be suitable to grow in the hatchery. Okay, so you have to do a testing here. That's why you do a research on digestibility of certain microalgae. It may have the desired nutrient, the desired energy, it may have the desired size and acceptability, but if it is not digestible by the growing larvae, then the larvae can starve to death also. All right, so remember this, digestibility. Then you have the supply of energy and nutrient required. So whatever food you give to your fish larvae, other than being available, pure, good size, acceptable, and easily digested, you want to make sure so your live food contains the required energy, nutrients, the fatty acids available for them, the protein available for them to grow and to multiply. Especially you want to grow them in your, what do you call this? In your containers. All right. Right. So there are quite a number of live food organisms which we grow. Okay. What are the types of live food organisms? And number 1 would be microalgae. Now, microalgae runs between 5 to 25 micron. Now, what is this micron? What size is this? Can somebody tell me how small is 1 micron, for instance? How small is 1 micron? Any idea? When I say, hey, that one is 1 micron, what does it mean? Can you imagine the size? Can someone? What does it mean? Macam mana kita nak, satu micron tu berapa? Macam mana nak pengukuran dia? Okay, kita tahu, we know 1 meter. Okay, 1 meter has 100 centimeter, right? And 1 centimeter has 10 millimeter. Alright, so in 1 centimeter, dalam 1 centimeter ada 10 millimeter kan? Alright. So, dalam 1 millimeter ada berapa micron? In 1 millimeter, how many microns are there?
1000.
1000. That's right. So there is 1000 micron in 1 millimeter. So if you have a microalgae, which is 5 micron, this is 5 divided by 1000. All right. So when you divided 5 by 1000, okay, let's do that. 5 divided by 1000, that is 0.005 millimeter, very small. 0.005 millimeter. You can't see with your eyes, you know. Okay. Even 25 micron also, 25 divided by 1000. That is 0.025 millimeter. You can't see that with your own eyes. They're too small. What you can see are very specks of powder. So kalau size 5 sampai 25 micron, you nampak dalam air tu, air warna hijau je. Lepas tu nampak macam titik-titik warna hijau. Okay lah. That is between 5 to 25 micron. That's about it. You can't see the individual cells. Alright? And this microalgae, which people culture in the hatcheries, are used to feed your bivalves. Bivalves are what? Bivalves are your oyster, your mussels, kerang-kerangan, kerang, yeah? Alright? Mollusks. Your shrimp, penait shrimp, what is penait shrimp? Penait shrimp, shrimps, which are normally marine shrimp, udang laut lah. Yeah, like your tiger shrimp, like your white shrimp, those are all penait shrimp. Okay? A rotifer. So you can give your macroalgae directly to bivalves. You can give them to shrimp. You can give to other zooplankton like rotifer, copepod, and sometimes you give them to small fish also. All right. And then you have a bigger size, 50 to 200 microns. Okay, these are normally for your crustacean and fish. All right. And 50 to 100 micron are rotifer. Okay. And then 400 to 800 micron, you have Artemia, Nopli, you have your copepods also up to this size. All right. And for a limited scale, you can even use your live food organisms in the form of Moina, Daphnia, and Artemia. So, which are the more popular ones? Macroalgae as live food organism, rotifer, Artemia, and nowadays a lot of people are trying to culture copepods. Right, macroalgae, the smallest ones, your rotifer, your Artemia nopli, your copepods. Now ask me, Chela, what about probiotics and what about bacteria? Do you grow them and feed to animals also, your bacteria, or these probiotics? Yes, we can consider probiotics as live food also, but that will be a special topic. I will cover that on what do you call this, bacteria or even yeast for that matter, yeast, ragi. But these are the more popular ones, routinely cultured for as live food for your fish larvae, your microalgae, your rotifer, your Artemia, your copepods. And then on limited scale would be your Moina, your Daphnia, and the bigger size Artemia biomass. Now, your plants can be your microalgae, as I said, aquatic plants, but we will focus on microalgae and animals, all this. Today, the four major groups of live food diets for your fish are your microbes, as I said just now, your yeast, your probiotics, right? We will cover this as well. Microalgae, rotifer, and brine shrimp, okay? And a bit of copepod, okay? So this live fish, but for the bigger fish, sometimes we give live food organisms. Of course, we do not cover this in terms of the practice of live food production, but we might invite some people, some of your some practitioner or some culturists who have grown some of this, you know. Like sometimes I invite some students who have grown some crickets to share with us his or her knowledge on how they grow crickets, how they grow tubifax worms, for instance, and so on and so forth. Now, there is a class of live food organisms which we also culture sometimes. This is normally cultured by people who grow betta fish, ikan plaga, and these are the nematodes. Okay, and principally the white worms or vinegar worms and so on. So nematodes are sometimes cultured by people who are growing or enthusiasts of betta fish or ikan plaga. All right. Sometimes they grow moina too. This one, moina. There's the moina. Right. There you go, moina. Okay, ikan plaga or betta fish. All right. This is an example of your macroalgae. Chitoceras is a species of diatom which is very popularly cultured for in marine shrimp hatchery. Hatchery udang laut biasanya menternak microalgae Chitoceras. Okay. And the chlorella species tend to grow in fresh water or brackish water, but you don't normally find chlorella in what do you call this, in full strength seawater. They prefer brackish or fresh water. And chlorella, you can find it normally in freshwater hatcheries. They grow chlorella for freshwater fishes. Chitoceras is more for marine. You do have some aquatic plants like this duckweed or lemna, but some people culture live and give it to species of fish like your tilapia and so on. But as I said, we'll focus more on this. Infusoria. Now, if you're not seeing this particular or heard of this particular term, Infusoria is essentially a collection of microscopic organisms in fresh water or in marine water, principally in the form of ciliates, like this. What are ciliates? Ciliates are zooplankton, which is covered by small appendages, hair-like appendages called cilia around the body. Okay, like paramecium, amoeba, and all these things. So very, very small phytoplankton, very, very small zooplankton. Okay, and this is also food for live food organisms for your fish larvae. All right, now in the lab, when we are ready with the face-to-face meeting, we will start to grow infusoria in the lab. All right. Now, whether we do infusoria culture before we start the face-to-face meeting, I will have to think about that, because I rather that you do it in a group. At least you can experience it in the lab, and you can watch it grow in your lab as well and observe under the microscope. Okay, this is what I was contemplating. I was really contemplating, whether I should give you a specific assignment for your lab. I figure out it might not be useful. It might not be helpful so much. But I rather that we do this when we do the face-to-face. Okay. Artemia, obviously, a lot of you may know already about Artemia. We culture Artemia. These are the Artemia adults. This is what we call as the biomass. When they're big in quantity, we call it Artemia biomass. This is Artemia noplii, which is very, very small Artemia just hatched from the cyst or the eggs. We grow this, especially the noplii, to give it to our larval fish. These are your rotifers, looks like this, all right. When we start to grow infusoria in the lab, you will see lots of rotifers. This is your Daphnia. Daphnia looks like this. Okay, in Malaysia, bahasa Malaysia, we call it kutu air, Daphnia. All right, or water fleece. Sometimes they call it water fleece. All right, and here are the eggs of the Daphnia. You see this? The eggs. That's the intestine. All right. Here, lots of eggs. This outside here, these are all the baby Daphnia. Right, the small ones. They look like the mom. All baby Daphnia. This is the eggs. Lots of eggs per Daphnia. See that? This is Moina. All right. Moina looks like Daphnia, but not as cute. People say, hey, yeah, so cute lah, the Daphnia. Okay, it is cuter, looks like it, you know. Moina not so cute, but Moina is very important. In fact, very important in many of our aquatic species production. All right. A lot of people grow Moina to feed to their betta fish, Moina. All right. Moina tend to be smaller than Daphnia. Daphnia is bigger. So that is why a lot of betta fish grower enthusiasts prefer to grow Moina. Okay. You will normally find Moina in a lot of this sewage water treatment plant. Okay. A lot of people grow Moina. I mean, they grow naturally there because the sewage treatment plant, the water is very rich in nutrient, you know, a lot of nitrogen there, phosphorus, all these things. A lot of microalgae growing, and this Moina feeds on the microalgae. They feed on the microalgae, they grow into large numbers and quantities in the sewage treatment plants. These are your copepods. Look at this. Copepods also quite abundant in your infusoria culture. When we do that later on. This is how it looks like. Now, copepod is the small members of crustacean. This is crustacean, you know, because it can undergo metamorphosis and change its what do you call this? The carapace. Okay, copepod boleh salin kulit untuk membesar. All right, and undergoes different stages. And this, you know, in the wild, in the natural system, copepod is very important live food for fish larvae. Okay, di luar sana secara semula jadi copepod adalah salah satu makanan hidup semula jadi yang paling penting untuk anak-anak ikan. Very important, alright. Mealworms, you see this sometimes in aquarium shops. They grow this to feed to the arowana, to feed to their big fish, to feed to their birds lah, and so on. Okay, so this mealworms actually is the larvae of a beetle. Okay, ini sebenarnya adalah larva beetle ataupun kumbang. Alright, larva kumbang ni sebenarnya, ya. Bloodworm. Okay, tengok ni bloodworm. Bloodworm also a larvae. So this mealworm is a larvae of beetle. Bloodworm is a larvae of a particular flying insect called midge or agas. Now, agas, AGS, agas in bahasa Malaysia. Alright, it is a midge, they call it in English. Now, this is the larvae, and this larvae, you can normally find it also in sewage lagoons at the base there, red in color, or in recirculating aquaculture system. You can find a lot of these bloodworms growing at the bottom of the tank. Alright, some countries grow this. Some countries grow this, like China. They grow a lot of bloodworm, export it out. Okay, and you can buy these bloodworms also in the form of blister packs or dalam, okay, in the kilo basis and so on. They sell it, okay, frozen as well. These are all microworms. You can, we will grow this in the lab later on, microworms. Okay, you can use this to feed your fish as well. Now, remember this microworm that we grow as live food is what we call as a free living microworm, free living. Free living is important because it does not infect people, it is not parasitic. a number of microworms are parasitic. Okay, setengah microworm ni, like your white worm, what do you call this? Tapeworm and so on. The one is parasitic, and for you it can cause disease to humans. But this microworm or this Panagrellus ni, dia bukan parasitic dan dia dipanggil free living. Yeah? They don't live in the bodies of animals or in people. They live on their own. Okay, in soil especially. Kat tanah. Sebab tu kat tanah tu, if you take the soil sample, you can find quite a number of these microworm as well. Crickets, people grow and give it to your fish. Sometimes they grow big frogs, also frogs given to arowana like this. Now you see arowana can be trained to eat pellets. Okay, now you boleh train arowana makan pellet, but some people just prefer to give live food, big fish, big frogs like this to your arowana. Now some fish like this, minnow, mosquito fish, are called feeder fish. Now feeder fish means what? Now feeder fish you can put in your aquarium, right? And it becomes food also for your bigger fish, for your Oscar, for your what do you call this? Siakap and so on. Okay, some bigger fish would prefer this, and these are called feeder fish. Crustacean, you have glass shrimp. Glass shrimp, this one in Bahasa Malaysia we call it udang gantung. Udang gantung, you can find it a lot. If you've been to Ladang 10 UPM, Ladang 10 UPM, in front of that, what do you call that place, where your students normally stay, Serumpun, in front of the Serumpun hostel. You go down to Ladang 10, you'll find that among the weeds or at the side of the ponds, you can get a lot of grass shrimp. Okay. Gamarus, fairy shrimp also you can grow them. Okay. And then this is also food for your fish lah. Polychaete. Now polychaete is sandworm. Is important especially for the marine shrimp hatchery. Now because it's so big like this, you don't feed it to your fish larvae, but you feed this to your broodstock. Okay. Polychaete ni kita bagi makan kepada induk udang. Induk udang suka makan polychaete. Ya. Tapi kita tak bagi dia pada anak udang lah, because dia besar kan. They're so big. You don't give it to the fish shrimp larvae. You give it as live food to your induk udang. Yeah, to your broodstock, shrimp broodstock. And it has a lot of lipid content, lipid dia tinggi, and then when you give it to your broodstock, normally the growth will be good. They will have produced a lot of eggs, okay, and so on. All right, so in general, your live food organism is very important. Even though we will focus more on live reproduction for baby fish, for fish larvae, we'll focus a lot on those plants and animals that we grow in microscopic scale, But remember that the bigger ones like this, like the polychaete, are also very important. Okay, bigger ones like the bloodworm are also very important, like the microworm. They're also important for the industry. Yeah, even though in many cases the general what do you call this? A lot of commercial production of aquaculture, we use the zooplankton and the phytoplankton as live food organism. Okay, jadi secara amnya beginilah ya senario lebih kurang tentang live food production, jenis-jenis live food yang kita guna. So then on after this, in our next slides next week and so on, we'll focus on specific live food organism. Okay, we can start with infusoria, we can start with microalgae, whatever, and then continue from there. Whatever we've gone through today. All right. Okay, I think I want to stop there here for the lecture. All right. Okay, do you guys have any questions for me? Ada soalan tak so far? What I've mentioned, and then apa nama tu? the issues, something that you're not sure of, something you want to share with me, or you want to give a comment, atau whatever. Go ahead. Or you might have experience growing live food, something else which I do not cover. All right. Anything? Anybody?
Saya rasa okey lah, tapi macam dia tak berapa berkaitan sangat dengan hari ni lah.
Yes.
Okay, saya nak minta pendapat Cik Ella lah pasal penggunaan Artemia as live food sebab macam mana experience sebelum ni dia macam banyak saya punya fry mati sebab dia cedera sebab spine tajam kan. So adakah dia okey untuk gunakan Artemia?
Okay, first of all, ikan apa yang you grow tu? What fish do you grow?
Sea bass.
Sea bass. You're growing sea bass. Meaning that you grow means you hatch it or what? You hatch your sea bass?
Yeah.
Yeah, you hatch your sea bass. Okay, now you hatch your sea bass where? Is it in UPM or somewhere else?
Oh no, kat ni tempat intern.
Okay, you grow your sea bass during your internship.
Yeah, yep.
Okay, and then what is the first food for your sea bass?
First food. Artemia.
Artemia, yeah? Now, this can be an issue also. I wonder whether the company where you did your internship, the sea bass that the first food they can eat your Artemia, no play? Dia boleh makan ke atau too big for their mouth?
Boleh. Boleh makan.
Boleh makan kan? Okay. So dia boleh makan. And then what did you find? What was the problem?
Macam mana mortality rate dia tinggi lah day by day. Macam hari ni setumpuk, lepas tu esok setumpuk. Elok ada lebih kurang 80,000 setelah jadi macam 8,000 dia tinggal. Macam tu lah.
Okay. Now, as I said, marine feed, marine fish is a very, very interesting industry, marine fish production, especially the marine level feed. Okay. And the live food organism is critical when you talk about marine feed. I found that, for instance, if you grow your, what do you call this? You grow your ikan kerapu or grouper. Now grouper, the larva is very small, and grouper, people don't give Artemia in the beginning. They give rotifer, right? They give rotifer, and then they put also green water. What is green water? Green water is basically your phytoplankton, right? So they grow the phytoplankton, and they provide a greenish background in the tank where they grow the animals, or they grow your fish. And then in that particular tank where they grow the fish, it's green in color. They also add the rotifer, which is small in size. And the rotifer tend to be eaten very well by your fish, for fish larvae. Now it could be in Hafizul, it could be that when you are giving feed to your live food, or your Artemia to your growing fish larvae, it could be that not all of them can consume your Artemia nopli. So quite a number of them are big enough to eat the nopli, but it could be that majority still prefer your rotifer, which is smaller than your Artemia. So this is a situation that we find in many cases where we grow, what do you call this? The larvae of ikan siakap or sea bass. Okay. So we had an issue also. We had some issue also with some people in Terengganu, near Kuala Terengganu before, where this guy grew ikan siakap, okay, or the sea bass in the hatchery. So he had some problem with his sea bass mortality also. So what we did was we encouraged him to make green water, air hijau. Okay. So first, after the sea bass has hatched, we asked him to put in the green water in the tank and grow some rotifer, and then feed your sea bass. And we found that when he do so, his survival of larvae is improved. Okay, so as I said, Hafizul, it could be that the size of your live food is a bit too big for a lot of the sea bass larvae. Yeah, so that's why you have mortality a lot in the beginning. Okay, now the Artemia is useful especially at the later stages after you feed them with rotifer. But in the beginning, rotifer is the normally preferred live food organism. Okay. All right. Anybody else?
Okay, thank you lah.
Sure. Anybody else have issues or anything want to share? Okay. If you see people growing marine shrimp hatchery, udang laut, the first food is normally the microalgae. And then after that, they switch from microalgae to Artemia. Marine shrimp, you don't give rotifer. See, that's a bit, it's a bit interesting, isn't it? So once we go to that topic on marine shrimp, I will share with you why they don't grow rotifer for marine shrimp. They prefer after microalgae, the truth, they just directly give the Artemia. Okay, but for marine fish like grouper or sea bass, they tend to give rotifer first. All right. Now, coming back to your class, okay. Atira, you ambil attendance kan, Atira? Atira?
Betul je lah. Dah ambil dah.
Okay, good. Thanks. So you take also attendance for together with the lab, because as I said, I joined together the class and the lab today. Okay, boleh Atira ya?
Okay je lah.
I just put lab and attendance for the class. Now, I was given the list already by Atira on the groups for the lab. Okay, we have 6 labs. We have 6 groups, and each group is about 6 students lah. All right, so we will not have any lab today. I will not give you an assignment, as I said just now. It's just early days of our class, and some of you are in kampung or already not yet here. I worry if I give you some assignment, it may not be fair because some of you may not have access to things that you want to get and start off with your lab at home and so on, a small project that I want to give you. Now, I understand that you all will need to be back in campus bila when do you guys need to be back in campus? Is it like early of November ke? Berapa? Anyone?
8 November, if I'm not mistaken lah. Week 4.
Week 4, 8 November. Yeah, the same. Okay, the rest okay, yeah. Is it like that? Yeah?
Yes, Cikgu.
Okay, so week 4. So week 4. So week 4 then I actually I think that week 4 will be the best time to actually start something more intensive on our lab. So in the meantime, between today and the next 3 labs, I will try to figure out something that you can do at the leisure of your home or in campus. I know some of you are in campus already. So that you just keep in touch with the live food production lab. Simple things to do. I may ask you to do some research a bit, or do a bit of reading on the especially on live food organism or certain topics, very simple ones. But we will do the lab when we have face-to-face meeting later on, so that it's more you can see more clearly. I just to me it's very important to actually be involved in a lab so that you can see everything clearly, and I can even explain to you better when the organism is in front of your eyes, right? You look at it at the microscope, you say, "Cik, what is this, man? It looks like a bear." What? A bear inside the microscope? What is it? So then there is actually an organism that looks like a bear, So look at that, something interesting, So and you do not know what it is, moving very fast, zigzag here and there. So these are the things which can only be experienced in the lab together with all of you. All right. So that's about it for today. And thank you for joining the class, everybody. And thank you, Atira, for taking the attendance, Atira. Yeah?
Welcome, Cik Lah.
Then you just send me through my email, kan. And then the rest of you, thanks everyone for joining the class. And then I hope you have a splendid or fantastic weekend starting tomorrow, Saturday, Sunday. All right. And by the way, for those of you who are taking my AKU4201, you will join me on Wednesday, right? Or you still join my class on Monday?
Yang Prof Muni, Dr. Muni tukar dengan Cik Lah kan?
Yes.
Dia bagi mention about your schedule, ni schedule.
Did she mention about the schedule?
Yeah.
Yeah.
Yours on Wednesday and hers on Monday.
Right. So then on Monday, you don't have your AKU3201. So your AKU3201 will be on Wednesday morning pukul 8. Alright? Betul kan? Yeah.
Betul, Cik Lah.
Good, Liza. So make sure, okay, make sure you know about that. So on Wednesday, it's like really a crazy day for me. Wednesday 8 to 10, 11 to 1, or 11 to what? 11 to 1, and then 3 to 5. So it's like, wow, one full day, man, with all of you. I'm okay with that. It's just that, alamak, boringnya tengok Cik Lah the whole day, man. Alright. Tak boring kan? Boleh kan? Boleh kan, guys?
Boleh je, Cik Lah.
Boleh je.
Boleh.
Let's have some fun. Okay, next week. Next week in the AKU 3201 on Wednesday. All right, next week, AKU 3201 on Wednesday. All of you will come to class in lab wearing some kind of a hat, topi. Okay, even yang pakai tudung tu kena pakai topi. Your favorite hat, I don't care what color. It could be even a Pokemon hat. Okay, I don't care. It could even be your panda hat ke apa, ada telinga tu. Okay, so everybody will take picture next Wednesday morning pakai hat. Alright, so itu you punya assignment. That's for AKU 3201 tau. And I will have my hat also. Alright, I will have my own hat also. And ingatkan ni, Atira, ingatkan boleh
Boleh, Cik Lah.
Yelah, okay. Kalau tak nanti boring lah kan. So let's have some fun in class, you know. Alright. Okay, so I just want to say that to all of you and have a good weekend and enjoy your weekend, alright. And come refresh on Monday morning together class dengan Dr. Murni. And then later on with me Tuesday or Wednesday like that. Okay, so that's it for today. Thank you very much and see you guys next week. Okay, thank you. Assalamualaikum and very good afternoon. Bye.